Uh, this lecture that I'm going to attempt to share with you, by its very nature, violates everything I've been trying to do since the dietary supplement industry was founded in 1994 with the passage of the Dietary Supplement Health and Education Act. And the reason I'm doing it is because surely with various publications, television, on your cell phones and computers, you're getting all this information about conflicts that are recurring with people charging that there's been politicizing of scientific information, and that people have agendas that they support any data that they can find that supports that agenda, and they ignore any information that doesn't support their agenda for what they're trying to promote. I hope that you'll stay with me because, for example, I love to go to the symphony orchestra in Dallas or Fort Worth. I can't play an instrument on the amphitheater. I can't read music. However, I can appreciate the music. And I hope that as we go through this information that you may not understand or comprehend everything that's quite detailed that will be in this lecture, but I hope you can hear the music of how incredibly detailed the process of life, of where the genes in each of our cells take elements on the composition of matter chart, which is about 100, and there is absolutely no evidence or sign of any kind of life in any of these elements, yet our cells with the code that is in nucleic acids or our genes or DNA can take these elements and hook them together, and behold, we have life. And we'll go through some of this process that I hope, again, that you'll understand or hear the music, but much like when I go to the symphony, I can't play any instrument or could not read any of the music that produces the sounds and harmony and beauty that comes from music. So with this, we'll start with the first slide. You may or may not be aware of that in the year 2020, there was an open letter published that 10,000 medical and scientific individual professionals signed the Barrington Declaration. Now, this was carried primarily in the conservative media, but the national media pretty well dropped it because they did not want the attention brought to the fact that these thousands of scientific and medical professionals were protesting the politicalization of the coronavirus pandemic, and also details about the problems of the RNA shot and various pandemic measures of 6 feet and everybody wearing a mask and other things that in the past that scientifically had been shown that they didn't do much. And here they were enforcing this with mandates, demanding that people do it, and the public media has been promoting this. With time, this proclamation is the efforts have been made to kind of expand it to include the politicalization of climate change. Now, there's no question that the climate's been changing as long as I've been around, which will soon be 90 years. But saying it's due to industrialization or the efforts of man becomes a very interesting question. And as I'll point out, those people that have questioned this because it's not part of the political agenda have been penalized. And this Barrington Declaration now If you check, it's close to 1 million scientists and medical people have signed it, all protesting the politicalization of scientific information. And they sick and tired of, quote, the political agenda or the story of the political party in power and the media that seems to be their spokesman are developing an agenda about the pandemic and also about the climate change and penalizing the oil and gas industry and anything that's a source of energy of the past and promoting solar and wind energy. So we'll go to the next slide. A good example of this, you might say, well, does politicalization of science matter? Well, I was watching television, and on one of the conservative channels, they brought out a segment that a community college professor that had been teaching biology for 20 years in San Antonio, Texas, in his lecture on chromosomes, showed on the screen that in the 23 chromosomes, chromosomal pairs, there's a total of 46 chromosomes, but they're in pairs, that in the 23rd position, if you have 2 X chromosomes, that's the karyotype for a female. And all the characteristics hormonally and physically that we associate with females are associated with this XX pair of chromosomes in the 23rd position. However, he also showed a karyotype, which is all the chromosomes laid out. I'll show you an example here in a minute. That an XY, and the little y is just a tiny portion of the X, that if you have an XY, this is the karyotype of a human male. Well, 4 students in this lecture stood up, walked out, filed a protest with the university administration. And what was their beef and what is their agenda? Well, they said that sex is determined by one's feelings and socialization and personal preference. And this is what produces males and females, which is the agenda that's being promoted across the country in education. Well, allegedly, the teacher was fired for promoting religious dogma. And they ignore the science that was presented that's been available for decades. In this case is on appeal. So this is just one example of how ridiculous politicalization of science can become, where truth is trashed, even when it is well-established scientific fact. Now, this is what the professor showed, and to the right, you see a jumble of chromosomes from one white cell, where you have added a fluorescent dye so that it binds to all the nucleic acids or the DNA, and they show up, and you take a photograph of this jumble of slides from one white cell, and every cell in the body has this in the center, and you lay it out to the left from the longest in the number 1 position to the shortest in the 23rd position, which you see in the yellow block. And this is the karyotype of a human male. If you had 2 X's identical laying side by side, it would be a female. This has been known for decades. And I show it to you because this is hard, well-established science that by political agendas, this professor got fired, and one can only hope that out of this legal conflict, that truth will prevail. Now, the effort to expand from protests about the coronavirus, then where it came from and its and the problems of the RNA shot. And I will not call it an RNA vaccine, which I will get into and show you why. But they've tried to expand this, that there is politicalization of climate change. And in this, the geological history of the continents before the industrial age is ignored, of times when it became very dry and wet, and times very cold and very hot, totally unrelated to the Industrial Revolution. And that the fact that people that are promoting this agenda look for data and sites where they collect information that supports their propaganda or their agenda, and they cite this. However, there are data can be gathered from sites that do not support this agenda, and those are ignored. And in fact, a professor at the University of Miami and Miami, Florida, published a paper pointing out this and giving a balanced presentation. And his peers caused so much furor that buy into the agenda that he lost his position on the faculty at this university. So this is not just a theoretical thing. This has actually happened. And what it is that open and balanced debate is silenced, not only on the pandemic, but on the climate change and on the new form of the vaccine type technology. And out of this, there are actually federal mandates demanding that citizens do certain things and don't do other things. And in fact, the oil and gas industry has been terribly penalized. And we've gone from a country that export energy to one that has to import energy because of federal policies against oil and gas industry. Even the gotten into major sources of funding to block loans for companies that want to develop oil and gas. And of course, the price of gasoline has markedly been increased by this. But as you look on the highway and see the number of trucks that are absolutely essential for moving goods across the country, that God only knows what's going to happen with the supply of diesel, which these trucks burn huge amounts of, and it comes from the oil and gas industry. And of course, the solar and wind industry is being promoted with tax funds, is because that is positive in this agenda that we're living under. Now, the coronavirus 19 issue is more complex, and that's where in this lecture that I'm afraid that the details of it are, because so much in science is based on words and phrases that come from Latin and Greek, and we don't use them in common everyday language. And so the layperson that has not had, say, multiple courses in biological and scientific science don't know what you're talking about. And that I want to point out that Robert Redfield, a medical doctor who was head of the Communicable Disease Center, made a statement early in the pandemic, and I quote him here: "I have analyzed the viral nucleic acid sequences," and that's the DNA in the virus, and that the changes from the wild strain to the pandemic strain do not reflect a natural mutation. He went on further to say that the DNA sequences are more in keeping with human manipulation. And for this statement, he was silenced, and the national media just dropped it like a hot potato, because they didn't want an expert that had been in the field of virology for decades questioning the source of the virus that may or may not have come from the Wuhan laboratory in China. Also, you have found it's science that has been suppressed, that a former Wuhan lab technician said, we worked on contract with natural viruses on an American NIH, National Institute of Health funding grant. Now, this was initially denied by Dr. Anthony Fauci, but under the Freedom of Information Act that allows people to go into the records of the federal government, it was found out that this was a bald-faced lie, and that hundreds of thousands of dollars had been given to this laboratory in China, because I won't get really much into it, because the NIH has been financing, and the Department of Defense over the years, different projects for biological warfare. And under the Freedom of Information Act, this has been uncovered. And so they've gone to great lengths to try to find ways to do the research they want to do without being able to be understood or found by the citizens of the country that are actually paying the taxes for this many people feel like onerous activity. But there was no question that they financed research to alter wild virus genes found in animals in this laboratory under an NIH grant. Now, because of the problems that have come from the RNA technology, which I'll briefly get into, that for over 50 years, the Center for Disease Control had a definition, and I'll quote to you, quote, a vaccine is produced by a substance that prevents an infection. Well, this new technology doesn't prevent an infection. So for a politicized definition, the CDC changed the definition of a vaccine to a vaccine is produced by a substance that stimulates the immune system. And I might add, the evidence to date strongly suggests that it confuses the immune system with this new technology. And I hope before we're through, you will understand in the process of how cells work. I had 2 years working on an academic degree in cellular physiology, which is a big words to say, how do cells work? Individual cells in the plant kingdom and individual cells in the animal kingdom, of which human beings are a part of. We have trillions of cells, and they all have to work together, and they all have incredible systems in them that time and time again, I have asked, how in the world did this ever come to be? So detailed and such design. And of course, there are those with the agenda out there in the media and in education saying there is no intelligent design. And I'll leave that up to you as we go through some of the, just some of the information about how cells work that I think is very important to know, to understand or comprehend how there could be so much controversy and the issuing of the Barrington Proclamation of scientists and medical people protesting the politicalization of science. Now, let's focus a little bit on the RNA technology. And what is the RNA? Well, desoxynucleic acid, or DNA, is the code of life in plants and animals. And I was surprised to find out that the DNA in plant genes is exactly the same DNA that is in the animal kingdom. And that raises the question, how did this come to be? And I won't go into the experiments that have been done to show that you can insert certain gene segments from a human genome or karyotype that I showed you before into the plant kingdom, and that proteins and enzymes can be made by the plant that can be extracted, and they're just exactly like the protein or the enzyme that is made in the animal cells. Now, in the desoxynucleic acid, there are 4 nucleic acid molecules that make up DNA. And we use an A for adenine is one of them. And then we have a T for thymine, which is the second amino acid. And the third one is guanine, which we use G in writings to keep from having to spell out the word every time. And then cytosine, which is the fourth one. Now, the importance about this 4 amino acid molecules is that this code is much like what you hear about. There is a code in your cell phone or your laptop or your desktop computer, that this code is somewhere between 10 times and, shall we say, 1,000 times more powerful than the code that is in this cell phone right here that I hold up in my hand. And one of the reasons I know this is because of when you have a little success in science, there are always those that think you know a whole lot more than you do. And here in the Dallas-Fort Worth area, there are actually research labs trying to develop software and hardware will be the computers we'll have 5, 10, 15, 20 years from now. And I've had them come and spend a half a day with me wanting to know, thinking I know everything, how in the world an individual cell works at a temperature that we are about 98/16 degrees. And we have a cooling and heating system of our body that dissipates, keeps the temperature of the body at a pretty narrow zone. And it takes very little energy to run this computer code, which is part of, I would say, the symphony of life that I hope that you are beginning to appreciate, what I call the music of life, even if you don't follow all these details I give you. Now, but the DNA code is, as I say, the code of life, and it's in the nucleus of every cell, and there's a nuclear membrane around it. And the DNA can be considered much like the reference books in a library. If you had any education, you know that you may want information or going to have a test. That if you go to the library, you cannot take the reference books out of the library, but you can make a copy on a copy machine, and you can take it to your office or your room or any place you study or work. And in our cells, a copy is made from DNA inside the nuclear membrane, and this copy made is called RNA, and it's a little change in the sugar from desoxy to ribose sugar with these adenine, thymidine, guanine, and cytosine. And in the RNA, the cytosine is replaced by another molecule called uracil. But the RNA is a working copy that goes to the assembly line that is in every cell that carries on cellular synthesis of hooking together nutrient molecules to make bioactive compounds that run the biochemistry of life. And this is true for every system in the body, not only the brain function, not only the cardiovascular heart system, not only the hormonal system, not only your kidneys and GI tract, but this is true for all systems. And these nutrient molecules that are hooked together start with amino acids, and then fatty acids, and then there are the carbohydrate units that are saccharides or sugars. And then with this, we have vitamins and minerals and very special plant-made phytonutrients. That's not FIDO, but PHYTO, phytonutrients, the molecules that when you hook them together, you get a biological compound that has taken elements that have absolutely no properties of life, and behold, we start seeing the aspects of life. Now, when RNA is made from the DNA, there is a very special system where you have a adenine in DNA. In the RNA, you will always have a thymidine that hooks together or is made from that. It's called a complementary copy, where you have a thymidine in a DNA chain, you'll have adenine in the RNA copy. And as you go on down, you'll see where you have a G that guanine always attaches to the cytosine. And the remarkable thing about this is that I have them laid out in a straight line there. And oftentimes you see a helix in magazines or pictures when they show you chromosomes. Well, at 60 degrees temperature, you have a helix form where the maternal DNA sequence is wrapped around the paternal nucleic acid history of the male and the combination of the male and the female egg and the male sperm. And this is very intricate in terms of that the adenine and the guanine are one class of molecules that I'll show you called a purine, and that the thymidine and cytidine are called a well, I'll just show you with the next slide. I believe it is. Well, no, before we get there, that they have instruments that you put in a buffer the free amino acids with a high temperature polymerase, which is an enzyme that is used to connect these nucleic acids in the order that I showed you before. But at 100 degrees, the helix unwinds and lays out flat. And with the polymerase, the nucleic acids can bind complementary, as I showed you before, to the chain. And then you go back to 60 degrees and it goes back to a helix. And so where you had one copy of each side, now you've got two helix. Then this little machine for polymerase chain reaction goes to 100 again, and the chromosome chain unwinds and lays out flat, and there the polymerase enzyme and the free nucleic acids can complementary bind again, and you go back to 60 degrees, and where you had two, you now have four. And this is goes over and over again, 60 to 100, 60 to 100. And as you see out there, it goes, it doubles every time. And where you have only one chain in a sample, if this goes all night long, going from 60 degrees to 100 degrees, you can have 1 million target nucleic acids there that can be analyzed. Now, there's a big problem with this, and you may have heard in the media that the person that developed this polymerase chain reaction said that the test for the coronavirus that utilizes the polymerase chain reaction is fallacious. It cannot be used. And the reason is that if you have more than one chain, let's say you have maybe 6 different chains of viruses, the next morning you'll have 6 million different targets all in the same product, so that this little chain of a virus that goes through this process of doubling every time the temperature is changed, doubles it. Well, everything in there doubles. And so all of us have in our serum or in our cells multiple viruses. And so you don't have a pure specimen to start out with. And this gives you false information in terms of trying to determine if a person has the China virus or doesn't have it, because there is so much reproduction through this polymerase chain reaction. Now, the RNA made from the DNA is divided into 2 functional types. And one part of the RNA is processed in a little area of the cell called the nucleolus, and it becomes what is called transfer RNA. And you put a little T in front of the RNA to stand for transfer. And of all things, this RNA goes into the cell cytoplasm, the fluid inside the cell, and then the interstitial space between cells, and looks for and finds to and binds nutrient molecules that will be required in the assembly line to make the bioactive compound that the other part of the RNA, which is called mRNA, which is messenger RNA, that carries the instructions to the assembly line that's in every cell to tell it where and how to put the different nutrient molecules that come from the transfer RNA that are brought in right at the right time, the exact nutrient that is recalled for in our genes. And I might add that you probably read that there are 30,000 genes. Well, depending on which one of those genes is activated to be expressed is what this little assembly line in the cell makes. In other words, it may make hormones, it may make neurotransmitters, it may make structure or functional compounds that are used in the body and used in the cell, or can be put into the serum and pumped around by the flow of the blood to the organs where it's needed. And I hope, though this is rather detailed, that you're beginning to appreciate that there's absolutely wonderful music in the organization and design of life, of how the nucleic acids work and function, and that this is solid, well-established science that has been established over the last 50 to nearly 100 years, that slowly, multiple laboratories in different countries have come together and published. And then not only do they accept it doing one, you've got to do it again in another laboratory to prove what was published in the first paper can be duplicated before there's acceptance to determine that this is hardcore, well-established scientific information. And that's where the Y chromosome and the 2 X chromosomes that I showed in the original karyotype came into being, that this is the karyotype for a female and the karyotype for a male, and that it's all in every cell of the human body, either the XX for the female or the XY for the male. Absolutely incredible detail, design, and engineering. And this is just to emphasize that in each cell, there's an assembly line which in this cartoon that's derived from a paper in Annual Reviews of Biochemistry, 1985, by a husband and wife biochemistry team that has over 300 references to published papers, that they put it all together to show you that in every cell there's this assembly line that I have big letters 1, 2, 3, which you don't have to recall. We're not going to have a test on this, but the first step is the ribosome, and primarily there, the RNA strip, the messenger RNA, the little mRNA, is read to tell you the order in which amino acids are put together to start a little chain of amino acids called a peptide, which is on the way to become a protein if it gets high enough molecular weight. Well, it goes to the second step called the endoplasmic reticulum, which is a membrane structure. And here the first carbohydrate is added to it. And what's the importance of this is the biochemistry of what is happening is in the center. And you see some round circles, and it looks like a little pitchfork there that it's 3 chains, and it's 9 molecules of mannose sugar. And the reason I have that there is because that is the principal molecule that is used in cellular synthesis that comes from the aloe vera leaf gel, mannose, acetylated mannose. And this makes a domain, which you can see on the surface of the cell over there to your left. And there are many of these on the surface. Fact is, we just have a few of them there because it gets like shag carpet and you can't even see them. But this domain is for cell-to-cell communication, because the trillions of cells in the human body must work together in harmony. And that's what this domain, made from the principal carbohydrate unit that's in aloe vera leaf gel, is assembled there in the second step of the endoplasmic reticulum. And then it begins to be modified as you go around, as it goes to the Golgi in step 3. And at the bottom of the cartoon, you have all the structure and function types of molecules that make up what this little assembly line can make inside of every cell. And before I leave this, I should point out that the cartoon of the cell that shows the nucleus and the DNA inside the nuclear membrane is what you can see on a light microscope at about 1,000 magnification. Well, it looks like in the cytoplasm around the nucleus, there's nothing there. But with proper stains and electron microscopy up to about 100,000 magnification, you can see the ribosome, step 1 in synthesis, the endoplasmic reticulum 2, and then the Golgi apparatus membranes is number 3. That is not the same magnification. That on 1, 2, 3 is about at 100,000 magnification. The cell to the far left is about 1,000 magnification. And of course, the activity inside of the biochemistry going on. And again, I'm trying to point out that the dynamics, engineering and design that goes on in every cell in the human body follows this pattern. And if you understand this diagram, you understand the power of nutrition, because depending upon which gene is activated out of the 30,000 genes, the biochemistry in the center changes. There will be different amino acids, different fatty acids, different carbohydrate units, different phytochemicals from the plant kingdom, minerals and all, to make up whether it's going to be a hormone or an enzyme or a structure in the human body or a functional molecule. That's important to know that that code that is in the karyotype that we started out runs all of this and is the information. And so the RNA that was made from the DNA is pulled through the ribosome to tell you which amino acids and how they're to be hooked together. Also, I attended a course in Washington, D.C. for a week to try to understand as much as possible about this. And from this point on in this course, they said downstream processing, because the details of how the biochemistry is modified that's in the center is really not well known. There are many papers being published trying to determine the entire process of going from a molecule that has no properties of life to be hooked together to make bioactive compounds that carry out the biochemistry of life in our body. But again, I don't expect you to understand or comprehend all of this, but I do hope, like I was talking about going to the symphony, that you appreciate the incredible music of the symphony of life that I'm trying to lay out to you. And we're just barely getting the basic information started. Now, this is a model of the cell, and that the energy to run everything in the cell, like that assembly line, comes from mitochondria. And also, you will notice a little red thing under the nucleolus, that the RNA that is made from the DNA, that is the transport RNA to go out and look for the nutrient molecules, is processed in a little area inside the nucleus called the nucleolus to become the transport RNA. And the cartoon had everything laid out in a kind of assembly line, but it's all kind of jumbled up in a cell. And remarkably, the cell knows how to do all this. And of course, the mitochondria is providing the energy that from breaking the bonds of sugars and between each carbon bond, which in the process of photosynthesis, where energy comes from the sun to hit chlorophyll, and with chlorophyll and carbon dioxide, which we breathe out and which comes from fermentation processes, these carbon atoms are hooked together, and a little bit of the energy from the sun is put in that bond between carbons. But the mitochondria reverses this process of energy collection from the sun and transfers a little bit of that energy to ATP, adenosine triphosphate, which is the wet cell battery that everything in the process of life is run on that energy. There's some incredible information that these mitochondria are much like what happens with your hot water heater in your home or your central processing unit for heating the house. It generates free radicals and nascent oxygen, which try to attach to anything they can find that it's like a magnet. If you've got a north and a south magnet, how they attract each other. Well, these free radicals, in the design of life on that assembly line, the body makes glutathione and superoxide dismutase, which are protective, and it's like a little cloud around each mitochondria to soak up or bind these free radicals to keep them from attaching to membranes in the cell, or even worse, to the nucleic acid or the DNA that's in the chromosomes of the process of life, because the order in which the molecules are put together in the DNA chain has information. If anything new is attached to it, like one of these free radicals, it changes the information. And the worst thing to do is called a mutation, which can end up being cancerous, that the cell no longer respects all the other cells and doesn't work together in cooperation. And these malignant cells will not honor the existence of other cells and try to take on their nutrients and occupy their space. And of course, malignant cells can actually cease the ability to be alive and cause cancer death. Well, this is just a little bit about the symphony of life and what goes on in one of the trillions of cells in your body, and how DNA is transferred to make copies that are RNA, and that there are two kinds of RNA, and that they function differently. Now, I was mentioning about the glutathione and superoxide dismutase. This is a slide given to me from Texas A&M, where they have a very special microscope that you have living cells in culture, and that what you see there, where you have in each one of them, is the nucleus in liver cells. Well, these cells are scanned at different wavelengths, and then they can make a composite picture of what glowed at the different wavelengths of this microscope that stores this information in the computer. And what they do is they put a antibody in the media that will attach to glutathione and to dismutase, and one of them is green, which is the glutathione, and yellow is the superoxide dismutase. And in fact, there's a little cloud of glutathione and superoxide dismutase around every mitochondria to try to soak up these free radicals before they can attach to anything else in the cell. And again, this is more detailed than I think that you generally would be interested in. But just like I say, going to the symphony, I appreciate the music, even though I can't read music and can't play any instrument on the stage. I'm trying to get across to you how beautiful the design of life going on in each cell in your body, and how it's detailed, and how it works, and why scientists are protesting people that know nothing about all of this come up with things like what is a male or a female is whatever you want to decide to be, rather than how your chromosomes came together at the time of conception. Now, this is an incredible display of truth about in the center, with the big round center there to the left, you see the nucleus. And what we have here is that the transport RNA, and this is a very special preparation where a silver layer was placed over the cell with radioactive transport RNA, giving off energy that's radiant energy, and it exposes the film and makes it black, just like on a mirror or in a black and white photograph. But it's all going on inside the nucleus. And then after it becomes transport RNA, it diffuses out in the cell, as you see to the right, looking for the nutrient molecules to bring to the assembly line that I have shown you. And this slide on the right, 88 minutes after activation of the gene, you can see that the T transport RNA is looking for the nutrient molecules in the fluid inside the cell, that hopefully in your diet you have provided the nutrient molecules that are required on the assembly line to properly assemble the bioactive compound. And I have a question mark there, because it looks like that the nucleolus which is the high processing area for the transport RNA, has moved out into the cytoplasm and is still making RNA. And that raises a question about what we do not know, but only this is done because of silver deposits by using isotopes that release radiation and expose the where the molecule is. And you can tell, and this is proof that the transport RNA is processed in the nucleolus and then moves out looking for the nutrients to bring to the assembly line that I've shown you. Now, the RNA's first thing I told you about was the information to tell the ribosome how to hook the amino acids and which ones together to start making the peptide or protein. The carbohydrates are added to the peptide in the endoplasmic reticulum in step number 2. And the number 3, the Golgi, is kind of like the finishing stage of an assembly line. And in Russian research behind the Iron Curtain, they were able to show that the trace elements, which are found in humate and are so low that you have to have a large amount of cells to even detect the trace elements, but the trace elements measure up to about 70, very small amount. And a good example that maybe you know about is that hemoglobin does not work or carry oxygen without its iron molecule at the center of the amino acids and the fatty acids and all, making a complex hemoglobin molecule, but you got to add the element. And this is a principle that's repeated time and time again. Now, I want to point out that the RNA quote unquote vaccine, which I want to call a shot because they had to change the definition because what they were doing doesn't prevent an infection, it just stimulates the immune system. That was a brilliant idea, and the reason it was a brilliant idea is that the RNA tells the assembly line how to make a protein. And of course, a protein is the antigen that the immune system can see to identify a foreign protein and develop antibodies against it. And what's not generally known, that in farmer vaccines, where they would take the entire length of a virus or a bacteria, that there are multiple segments of nucleic acid in there, for example, in the virus, to allow the virus to know how to transfer across the membrane inside the cell, and then instructions of how to take over the ribosome, the endoplasmic reticulum, and the Golgi, and get the cell to start making viral proteins and viral material instead of that which originally the cell was made for. Well, these whole virus vaccines, which were much more effective and actually prevented infections, also had a low incidence of actually causing the disease. For example, smallpox, which has an attenuated virus, in a certain percent, very low percentage, actually the people get the smallpox. And there's even a lower percentage that can die from actually receiving the vaccine. So the idea was to make take a RNA segment that codes for how to make the protein antigen that's in the vaccine, and never expose the host or the individual to the entire vaccine complement. And that way you would avoid anybody ever getting the disease, because you just make that one segment. Well, unfortunately, the fragment of the virus that the RNA is made from doesn't give you complete protection. And in fact, as we'll get into here in a minute, there's something else at work that caused a paper to be published that explained to me that I'm trying to get across to you on why this vaccine has been a catastrophe. It's not a vaccine, it's a shot. And I was at this slide saying this is the reason 15,000 scientists signed the Barrington political protest, that what I've been trying to share with you, these scientists know and understand, and we will soon get to something that is not generally known even among very well-informed scientists. And the paper that I'm talking about was published in Epic Times, May 3rd to the 9th edition. It was entitled "The Trojan Horse in RNA-Based, quote, vaccines," because we got it in quotes because it's a shot, it's not a vaccine. And the author was Kraus Stiegler, a molecular biologist for 30 years at the University of Gießen, had to go outside the United States and out of the political arena to get someone to really share this. And he had help from Andres Schnapf, Schnaph, who's also a molecular biologist in Germany, to try to explain to those that are interested why people would sign the Barrington protest and what the basis of it is. And they're not only what I've been showing you exists in each cell in your body, the assembly line and the information being brought in by your genes, and that your genes stay in the nucleus but makes a working copy that's sent to the assembly line. But there's an audit step biochemically that when the RNA is made from the DNA, it basically says, is this correct and complementary, and is it genomic? And what we mean by genomic is that is the RNA in its complementary sequence that I was showing you in one of the slides, like it was either in the maternal or the paternal genes. In other words, did it come from genetics from the mother and the father, that the RNA is properly made before it goes out of the nucleus to be used on the assembly line or to look for the nutrients. Now, when the transport RNA is made, that it's going to dictate how the messenger RNA utilizes the amino acids. There is a second audit step. And what this audit step in the nucleolus is this protein that's going to be made a foreign protein, or is it genomic? If it's genomic, it progresses to make that protein because it came from the mother and father genetic history. But if it's a foreign protein, it stops right there. It does not dictate the production of the antigen protein that would be hopefully effective to make antibodies against the virus. And by this, in the various products that you hear that different companies make, they add different things to this RNA shot to try to fool the immune system that even though this is viral or foreign protein, that it will go ahead and make it. And it seems to confuse the cell rather than produce the what you want. So the first step is called transcription where DNA is by complementary amino acid sequencing makes the RNA, and there's this audit cell. Is it correctly made, and is it genomic like it comes from the mother and father? And then the RNA is divided into two things, the little m RNA, which is the messenger RNA that gives the assembly line instructions of how to hook the nutrients together to make properly the bioactive compounds that run the biochemistry of life. And then the transport RNA that goes out and seeks nutrients and brings them into the assembly line, the proper nutrients at the proper time. And this is called translation to make protein. And there is an audit step here, which these molecular biologists are pointing out, which again says, is this protein going to be a protein that came from the mother and the father and was inherited? Or is this a foreign protein like from a virus? And at that point, if it's foreign, the production of the protein is blocked. And this is the problem with the RNA shot. And it's causing thing. And I might add that the federal government has a adverse reaction website. And the first time I was shown it about 2 years ago, there were at least 1,000 deaths from the RNA shot that was supposed to be a vaccine. Well, I was aware that if a patient is admitted to the hospital, that they say they have the coronavirus, that they try to support them to get over this. But if they have to go to ventilatory assist, that the lungs are affected so bad that you've got to have assist, the payment from the Medicare, Medicaid, and insurance companies go from about $15,000 to about $35,000. So there's an incentive to say this person's got ventilatory, very bad coronavirus. Well, with this in mind, I thought possibly this website of adverse reactions was padded. So I went into the individual patient stories of the so-called deaths from this RNA shot. And I was surprised to see that because they put a priority on people that were working in emergency rooms, nurses and physicians, many of them having to work double shifts to try to handle the volume of people coming in with the pandemic. And so they were among the first to get the new RNA shot. Well, they gave them the shot, and the history was they never came back to work. Their health collapsed on them, and they died in about 2 weeks and never came back to work. Over and over again, this was what was in this adverse reaction site. And now it's several thousand deaths have been caused, and you don't read about that in the national media because this does not fit the politicalization that the RNA shot is a great success and that everybody ought to have it. And if you don't have it, you can't fly on an airplane. You can't go into certain countries. You can't go to certain places unless you can prove that you've had the RNA shot. Well, this is part of what the protest of the scientists with the know of what I've been trying to share with you, which is, as I admit, is awfully detailed science, but I hope you appreciate the music of the details of how incredibly detailed our cells work and why the RNA shot has been a catastrophe, that it doesn't work as the idea was for it to work. And it's been a major problem. And in fact, the products made that they add different products to the RNA shot to try to fool the immune system to go ahead and make some of this foreign protein, that there are white cells called thymic white cells, identify these cells producing the viral protein that are unhealthy, and the white cells attempt to destroy them. And there's a process among our cells that it's genetic already called apoptosis, the activation of a program for destruction of these T cell white cells to destroy the efforts of the RNA technology to get the immune system to make this foreign antigen, which the cell is set up to reject. Then in the B lymphocytes, which are primarily in the lymph nodes and make antibodies, don't properly comprehend what is happening immunologically, and they don't bind to coronavirus cells, which is the purpose of the vaccine. And this is why the CDC had to change the definition to match the political agenda. And as I said before, for decades, a vaccine was defined as a substance that prevents an infection. The new definition is a vaccine is a substance that stimulates the immune system. And this authors from Germany try to point out it confuses the immune system, and it's a catastrophe. Now, one of the reasons I know something about this is that in the early days of the research for the active ingredient in aloe leaf gel, 7 patients from 7 different states contacted a little company about 10 minutes from my office in Grand Prairie and said, I was on my deathbed waiting to die from this new disease of unknown cause and no treatment called acquired immunodeficiency syndrome, or reduced to, let's call it as AIDS. Well, in an effort to try to do something in a hurry, there was a research virologist by the name of Jasbir Cologne, PhD, at the Southern Research Institute in Alabama. And she was quite famous because the National Institutes of Health sent her about 350 different compounds to search for something that would attack the AIDS virus. And she came up with AZT, which is a variation of thymidine that interferes with the metabolism of the AIDS virus. And in fact, it would stop the AIDS virus, but she did not recommend AZT because it was so toxic, it killed the cells that the virus was living in. And even though, well, what was happening in the public sector is that AIDS patients were chaining themselves and using handcuffs to the lead into the CDC and the FDA saying, you're trying to kill us. And so the Reagan White House said, give us something, and give us something now that's effective against the AIDS virus. And so they came out with AZT. Well, a research project was done in over 300 patients in Europe, and they were able to show that patients that got absolutely nothing for treatment live longer and had a better quality of life than if you gave them AZT. And it rapidly, just like Jasbir Cologne's research showed, was dropped in their other things. However, Dr. Cologne was in graduate school with one of the professors at Texas A&M, which was the #1 academic institution working on contract for Carrington Laboratories. And through this person at Texas A&M, we were able to get the active ingredient from the aloe, ace-mannin, or acetylated mannose, to Jasbir cologne, which she went through all the experiments that she did for the NIH to find AZT. And lo and behold, this is data from that she published in the journal Molecular Biothermy, Volume 3, in September 1991. And you can see that in the table that there was 98% inhibition of the HIV-1 virus in the RNA step that we have outlined before, that the active ingredient from the aloe was very effective, and it did not kill the cells that it was in. And this to the right is a graphic presentation of what you see in the data over the left. But the control virus proliferates extremely well. But if there's presence of acetylated mannose, that the production of the virus is greatly reduced. And only because of this did I feel like that I could begin to try to share with you why so many scientists and medical people are protesting the politicalization of science. And Dr. Cologne, in her paper that was published, in A here, you have the cells in culture, that there's no virus, and you just see a uniform cell distribution. If you introduce the virus, you see these blotchy areas that are called syncytia, where the cells fuse together, which is viral cytopathology, and it alters the cells. And however, if you add some of the active ingredient from the aloe, which was done in the cultures in number E, note again how nice and uniform the cells are, and there was a 98% plus inhibition of virus, because in the audit step in the RNA processing in the nucleolus, the virus is identified as a foreign protein, and so virus particles are not made. And this is an enhancement of the normal function of RNA in the nucleolus that is the basis for why the aloe oligosaccharide was effective against the AIDS virus. Now, Carrington Laboratory tried to use this data to advance it in the new drug approval process. However, they found out, just like Jasbir cologne showed, that there was absolutely no toxicity of the active ingredient of the aloe against cells. Well, the father of pharmacology, Western pharmacology, was named Paracelsus, the most noted person in world history from Switzerland. His most famous statement was, all drugs or all pharmaceuticals are poisons. The benefit depends on the dose, end of quote. Well, the active ingredient from the aloe would not kill any mice, rats, cats, dogs, and finally, even in undergraduate students at the University of Texas, and sterile IV ran into medical students in Houston. And that is pharmaceutical heresy. No toxicity. We could not give them an LD50 about how much of this stuff does it take to kill half the animals, and how much of it, the LD100, to kill all of them. There must be a wide window, or you can't proceed to the second step, because, quote unquote, all drugs are poisons. Because drugs interfere with the engineering and design of life to reduce symptoms, but do not address the cause for the symptoms are problems that the patient's having. However, the genes are programs to try to move the biochemistry and the physiology to the normal zone or homeostatic zone, as it is called by the father of human physiology, Dr. Hans Selye. This balance within cells is called homeostasis, which you see in slide E here, that the virus was not transcribed to make the viral protein because of the audit step that's in the nucleolus in the formation of transport RNA, where it transcribes into making the protein. The other thing is that we found out that it did not matter if a person had a deviation from normal that was physical, which we frequently call diseases or syndromes for the profiles, so we can discuss these deviations from normal in medicine. Also, that we found out it didn't matter if it was a physical illness, a mental, or an emotional illness, that by adding this to the diet, there was a significant incidence of improvement in humans. This, again, is pharmaceutical and medical heresy. I did not realize it before getting involved in this research, that medicine is organized one drug, one disease. You may not have focused on this any more than I did, but we don't use antibiotics to treat depression. We don't use psychotropic drugs to treat pneumonia. In fact, is in antibiotics, it is so one drug, one disease oriented that it depends on where you find the infectious organism, you use a different pharmaceutical drug. And so we had two strikes against the application to make the active ingredient from the aloe vera plant, acetylated mannose, a drug, because it was non-toxic and everything improved when you added it to the diet. Well, fortunately, in 1994, the Dietary Supplement Health and Education Act was passed, and we realized with all the scientific data that 12 different academic institutions had generated, that we did not have a pharmaceutical that acts or has a mechanism of action like a pharmaceutical, that it is utilized by the cells to move to the homeostatic or normal zone to support the normal biochemistry design and engineering of how cells work. And so if I had not been involved in the 1990s with research with Dr. Cologne, I don't think I could even begin to understand or appreciate what I've been trying to share with you about why there is so much confusion, protest, and upset in the scientific and medical and public sector, and the fact that the media and the predominant political party has politicized science to follow their agenda. And I hope with this presentation, though you may not have followed all the details, that if nothing else, you appreciate the incredible engineering and design of how cells work and how it can be used for good to improve and support an improvement in health. So with that, I'll bring it to end and hope that this has gotten across to you, much like when I go to the symphony. I can't play the music, I can't read the music, but I can understand and appreciate the beautiful outcome of what is presented by the symphony orchestra. And I hope you see the beauty and the symphony of life and how the genes are organized and run the biochemistry and the assembly line of life. Thank you. That's the end of this lecture.